Myself, I'm Sanket Desai from Dr. Amit Dutt Laboratory. At the outset, I would like to thank the organizers for this opportunity to present our work at this conference. So, for the today's talk, I'll be talking about the role of Fusobacterium abundance, which we found that it is associated with inflammation, poor survival, and other clinical features in the HPV negative tongue tumors. Yeah, talking about role of pathogens in cancer. Uh, this is a 2014 report which talks about nearly 15 to 20% of the cancer associated risk factors. You find that there are pathogens which are mainly either the causal or associated risk factor with the different tumor types among all the others, and which accounts to nearly 1 in 6 patients that can be attributed to infections. The burden of the cancer cases that we see is nearly 2.2 million cases worldwide. Now, most of this burden is actually from the developing countries, and so as we can see that the different oncoviruses and bacteria that we see to be associated with cancer, 80% of those cases are coming from developing countries. Now, it's not only the association, but at molecular level also, different studies have shown that specific pathogens they affect or they interfere with the molecular machinery. So here we see that the different hallmarks of cancers being changed, altered by the presence of specific bacteria and viruses. And this study is primarily that I'm highlighting is primarily from the colorectal tumors, but the same is true for most of the tumor types that we know. So the this sets the base for our study that we need to develop better pathogen detection methods, which are going to play an important role in understanding of the cancer. And our laboratory mainly deals with the next generation sequencing data sets, which is a multidimensional, which gives multidimensional aspect to the tumors. We understand different processes that are going on through the NGS dataset. So our the main aim of this particular study was to identify the pathogens from the sequencing datasets. So as to do so, we developed something called as infectious pathogen detector. Now, this infectious pathogen detector, I won't go into the details of the steps which underlie the complete identification process because that is not focus of today's talk. But what this particular pipeline or the tool does is basically identify the pathogen traces among it identifies pathogen traces among the reads or the DNA and RNA that is sequenced, and finally gives us the quantification, which is a normalized quantification for particular tumor samples. Now we use the same pipeline to analyze SARS-CoV-2 earlier, and that has been reported in the public domain. So as to study the tumor samples to create a landscape of pathogens from mainly the Indian sequencing samples, which has never been reported before. Our lab houses these nearly 357 samples, the NGS sequence samples, DNA, RNA included both, which are representing these 6 tumor types. To complement our study, we also use TCGA samples, which is a huge repository of RNA as well as DNA samples. Here, from there, we only use the RNA-seq samples, taking the tally of our analyzed samples to nearly 1,400. For today's talk, I'll be only restricting to the RNA-based analysis that we did because of the brevity of time. So here is a complete landscape of pathogens that we generated, which was mainly this included all the Indian samples as well as the TCGA samples, which represented these 4 tumor types. Now, first, from the data set, we had to look for the positive markers that we already know are associated with specific tumor types. So here we found a very high abundance, nearly 90% abundance of HPV in the cervical tumors. Secondly, going to the colorectal tumors, we found that the three primarily associated pathogens, which are Fusobacterium, Bacteroides, and pathogenic E. coli, are also abundantly present in colorectal tumors. Now, these all have been identified through our pipeline IPD. Coming to the head and neck tumor, which, as you just heard my colleagues talk, our laboratory focuses on head and neck tumor. Looking at the head and neck tumor, we found an interesting exclusivity between the two pathogens, HPV, which is mainly present in the Caucasian population, and the Fusobacterium, which is present across the Caucasian as well as the Indian population. So, just to summarize the prevalence of the two pathogens that I'm focusing right now, the HPV prevalence was found in around 90% tumors, and nearly 14% of the Caucasian tumors showed presence of HPV. Uh, and the presence or the prevalence of Fusobacterium in colorectum as well as HNSC was nearly comparable. Now, colorectal tumors are the ones wherein it has already been associated, shown, and proven to be involved in tumorigenesis as well as therapy resistance. So, just to summarize this particular plot in head and neck, we saw that the HPV and Fusobacterium prevalence was, or the occurrence was exclusive to each other. And what we wanted to do further is actually understand that how this HPV positive and Fusobacterium positive tumors are distinct from each other, because that is what will give us the molecular features which can be associated with Fusobacterium. So primarily we did a differential analysis between the samples that I just showed you, and we found that the Fusobacterium high group had a distinct gene signature of 60 genes, which was not present in the HPV high group. Now, this most of these genes are present, or they represent the inflammation pathway.
Dr. Sanket, can you please conclude your talk?
Yes, yes, sure. So we also found that there are genes which are down-regulated differentially in the two pathogen-associated tumors, and this we further validated the inflammation markers. We further validated in the Fusobacterium high group from the in-house tongue tumors. We did similar validations for the microRNAs that we identified. Primarily, these three microRNAs that are also involved in inflammation are also associated, interestingly, with the Fusobacterium abundance. It is not only molecular markers, but the immune signatures, and the three immune signature which represent the innate immunity are also found to be higher in the tumors which are Fusobacterium high. So that shows that it creates a pro-tumorigenic environment, and that is what is seen in the clinical data as well, in which from the in-house tongue tumors, we see that the Fusobacterium high tumors are surviving poorly. The same trend we saw in the TCGA tumors, when we only take the tongue tumors, they are surviving bad with Fusobacterium high abundance. Clinically, we also saw that extracapsular spread was also associated with the high Fusobacterium abundance. And as my colleague just presented a story on MMP10, we also found the association between Fusobacterium and expression of MMP10, which is a marker for nodal metastasis. So as to summarize, we developed an automated tool for NGS dataset analysis, and this is the first in-depth systematic analysis from Indian samples, the pathogen analysis that we present. We identify a subtype of HNSC tumors which is exclusive to HPV positivity, which has a distinct clinical and molecular features. And it not only shows different molecular features, but also immune features, as well as it shows association with extracapsular spread and survival in the tongue tumors. So with that, I conclude. I thank my guide, Dr. Amit Dutt. Very important clinical data shared by Dr. Sudhir there and Dr. Kumar Prabhash, which has led to the study. Our data collaborators, Dr. Murli Basham and Anil Banerjee, and our project contributors, lab members, and my institution for giving me the support and the fellowship to the study. Thank you.